Okay, my friends, welcome back. So part of today is going to be a survey through additional lipid molecules, some that are important in membranes, others that are important in pain and inflammation. Of course, you don't need to memorize structures. The point of this whole section of the class is that the structure of the molecule is related to its property, determines its properties and functions. So I'm not going to ask you, for example, which of the following is linoleic acid or something like that. But I might ask you, of the following fatty acids, which do you expect to have the lowest melting point? So be able to reason through with principles that we've talked about, but don't worry about memorizing structures. You're going to find in chapter 25, as you read through, there aren't actually even that many good practice problems in the first part of the chapter, because again, it's mostly about this molecule is here and it does this, and look at its structure, ooh, isn't that great, and then move on. So we're going to do some of this so that you have an idea in future chemistry and biology classes what these biomolecules actually are, because in some cases, if you don't see the atoms here, you're never going to see them. And so I want you to be aware of them, but we're going to be focusing on the principles that connect structure to function. So first, phospholipids. These are phosphoracylglycerols. You remember that triacylglycerols are, triglycerides are molecules where you have this 3-carbon alcohol glycerol, and each oxygen is involved in an ester with a long chain carboxylic acid that we've called a fatty acid, okay? Now let's, just a brief review at the beginning before we deal with this phospho thing. You've recognized the carboxylic acid before and how you can take an alcohol and react it with a carboxylic acid to give yourself an ester. Right? You recognize an ester. It turns out that phosphoric acid can also form esters. We mentioned phosphoric acid. I don't know, maybe you've encountered it in stuff about acids and bases. The first pKa is around 2, and the second, I think, I actually forget. But one of the pKa's is around 7. So the dominant form of phosphoric acid at physiological pH is probably that. You can add an alcohol to it in the same way that you would add an alcohol to a carboxylic acid. And what you get then is what we call a phosphoester. Okay? Same basic thing, right? Oxygen bonded to the carbonyl carbon. Oxygen from an alcohol bonded to a phosphorus. So by analogy, that's called a phosphoester. But it turns out that phosphoesters can undergo a second round of modification of ester formation to give you something called a phosphodiester. This is important in the structure of phospholipids and also is important in DNA. But the idea between both phosphoesters and phosphodiesters is that you've replaced some of the original OH groups of phosphoric acid with alcohols. Okay? So we're going to see a phosphodiester in phospholipids. Because to make a phospholipid, we basically take a triacylglycerol conceptually and remove one of the carboxylic acid groups. And then we make a phosphoester out of it. So let me show you the basic structure of a phospholipid. And the identity and length of the hydrocarbon chain on the fatty acid varies. 2, 4, 6, 8, 10, 12, 14, 16, something like that. So we won't worry necessarily about how many, 10, 12, 14, 16, how many carbons are in the chain. But then the third alcohol is the one that's involved in a phosphoester. Okay? So if you just have an OH group here, this is called phosphatidic acid. And do you see the phosphoester linkage between the alcohol of glycerol and then phosphoric acid? Okay, that's phosphatidic acid. Sometimes people will abbreviate that as PA. That's not found very much in eukaryotic cell membranes because we will actually replace the OH group in phosphatidic acid with another alcohol. But the general structure of this molecule involves what we will call a polar head group and then two nonpolar tails. And for the most part, in future classes, you're going to see all lipids represented schematically like this. We'll talk about that general shape in a minute, but I want to give you a sense for what other groups can be here. Okay, so alcohols that replace the OH group. The first is ethanolamine. That's just OH and then an amino group, which at physiological pH would be protonated, such that if this were in our phosphatidic acid, maybe I'll just draw, copy, paste, this would be the group attached. So we would make an ester between this alcohol and this phosphatidic acid. This lipid is called phosphatidylethanolamine. And one interesting feature is that it is neutral, though zwitterionic. So the head group is polar but has no net charge. That can be a way that of distinguishing some cell membranes from others. Remember when we were learning about antimicrobial peptides, I told you that the ability of a positively charged peptide to associate with a negatively charged membrane was important. But I also said while you're lysing bacteria, you also don't want to lyse your cells. And one of the ways that you can be selective against your cells, against bacteria versus your cells, is the fact that many of your cells actually have neutral head groups. Okay? A related head group is called choline. So this alcohol is the trimethyl version of ethanolamine. So this one is permanently positively charged. I don't intend to actually draw the same phospholipid every time. Are you comfortable in your mind taking this alcohol and putting it there? Yes? Okay. So that will be called phosphatidylcholine. And people that do a lot of stuff in lipids will call this PE or PC. There are some other ones that show up, but I'm not sure. Another common one is for the alcohol to actually be serine. So here's the OH group of the serine side chain. And then here's the NH3 and the carboxylate group. So if serine were attached here, you'd have a negative charge from the phosphate, a negative charge from the carboxylate, and then a positive charge from the ammonium. And that would make overall this membrane, that would make this lipid overall have a -1 charge. That's often abbreviated as PS or phosphatidylserine. And there are others, but these are sort of the main ones that you will see. Cell types will differ in the composition of the membrane. They will differ in the identity of the hydrocarbon chain and the degree of unsaturation. Some cell membranes will have double bonds there. That can influence how fluid the membrane is. And then sometimes the inner leaflet of the membrane that faces the cytosol will have a different composition than the outer leaflet that faces the extracellular matrix. And how all of that gets arranged, I don't know. That's sort of beyond the scope of the class. But these are the kinds of things you could encounter. Let's talk about shape, because this is a principle that you should be able to understand. What is the difference between a phospholipid that can form a membrane versus a detergent or a soap that forms a micelle? So notice that the phospholipid, as we've said, has a polar head group and then 2 nonpolar tails. Whereas a detergent or something that forms micelles would be like our long-chain carboxylic acids, right? In other words, that's terrible. But an example of something that would form a detergent might be like oleic acid or something, a long 18 carbon chain or something like that, where you have a polar head group and again, sort of the nonpolar tail. So schematically, let's draw that. And this is something you may have heard before. I can't remember whether I told it to you or not. If you look at the shape of a detergent versus a phospholipid, a phospholipid has a more cylindrical shape. I don't like those tails. I need to try again. Sorry. Has a more cylindrical shape, whereas the molecule that forms a detergent has a more sort of conical shape. Now, remember that these things operate in water environments. And remember that the hydrophobic effect means that it's energetically favorable for the nonpolar portions to be not exposed to water. So the detergents will self-assemble into a micelle, and the conical shape generates enough curvature as they associate with each other side to side that you can actually form this shape where all of the, in a single layer of detergent molecules, you can form a sphere where everything on the outside is polar and everything on the inside is nonpolar. And it's that conical shape that generates that curvature. And so this is a micelle. And the reason it works as soap is because it solubilizes the greasy things that we get on ourselves and puts them in a nonpolar environment inside the micelle, and the environment outside is soluble in water. In contrast, as the phospholipids assemble side to side, their cylindrical shape means that there's not much curvature. And so they will associate this way to sort of get their nonpolar tails out of water, but there's not enough curvature to really create a micelle. And so what will happen is that one leaflet will actually self-assemble with another in a way that puts their nonpolar tails in the middle, and this is your lipid bilayer, where if this is the cytosol and then this is the extracellular matrix, you now have a barrier. And it's an interesting barrier because anything that crosses the membrane has to get through polar environment, non-polar environment, and then polar environment again. Membranes are really important. It's what separates you from everything that is not you. And and within the cell, different organelles are separated by membranes and have different functions, different reaction vessels, so to speak. So this kind of self-assembly generated and directed by the shape of the phospholipid is pretty important. And of course, this would extend in front and back and side to side. Eventually, that lipid bilayer can curve back on itself, but the scale of curvature is a lot lower, and so you end up with much larger structures in terms of membranes. Okay, questions you want to ask about that? If you put this in water, will it spontaneously form a lipid bilayer? You can get it to do that. I think it can be tricky. There is a such thing as a monolayer, but that happens when you have just one thin layer of lipids at the surface of water, polar groups down, and actually, this is wild, non-polar groups out in the air. So that happens, and people do experiments that way. But yes, people do interesting experiments with membranes where they have they have a small pore that they then put a lipid bilayer across. And I don't know in terms of technical details how they what particular experiment they're doing, but you can do electrochemistry across a membrane like this that you make synthetically, and you can buy a lot of these lipid molecules. Okay, other questions? So there's a related molecule that's also found in membranes. Its structure, the atomic composition is different, but as we draw it, you're going to see how it very much mimics the shape of a phospholipid. These are called sphingomyelins. They're called sphingomyelins. The myelin part has to do with the fact that they are found relatively often in the membranes of the cells that form the myelin sheath around neurons. Sphingosine is an amine diol that looks like this. Do you need to memorize the structure? No. Do you need to understand why it can form, why it's comfortable in a membrane? Sure. Let's see. 2, 4, 6, 8, 10, 12, 13, 2, 4, 6, 8, 10, 12, 13. Okay, so like this, the molecule would be called sphingosine. Can you see the structural resemblance to glycerol, at least in this portion? You sort of have a 3-carbon chain with sort of groups on it, and there's a nonpolar tail here. So in the case of sphingomyelins, the sphingosine precursor comes with a nonpolar tail. And then the nitrogen gets modified as an amide with another fatty acid to make a second nonpolar tail. And I'm not worrying about how many carbons long that is. Starting to see the resemblance with the phospholipid. And then the head group will get modified by some of the same types of things as we've seen before with the phosphoacylglycerols. That is, you could put an ethanolamine group here. And this would be an example of a sphingomyelin. But hopefully you can see the presence of a polar head group and 2 nonpolar tails. And even though the structure is different, the bonds are different, it can assemble in a lipid bilayer just the same way. So sphingomyelins are sort of a different independent solution to the problem of how are we going to make a membrane. And in terms of why cells in the myelin sheath tend to have a lot of these, I don't know. But different lipids can change the fluidity of a membrane, can change how adaptable it is to changes in shape. Okay, so I think that's enough about phospholipids. There is a section of the text that talks about fat-soluble vitamins. These are hydrophobic vitamin molecules that can accumulate in adipocytes, which are the cells that store triglycerides. They include vitamins A, D, E, and K. And we could draw their structures here, but I don't think it's worth our time because it would just be, and here's this structure, and here's this structure. It would be useful to you to have seen these molecules so you have an idea of why they are lipid soluble. You can see that they're nonpolar. We will come back to vitamin D in the future. Sort of one of the last things we do is going to be how your body synthesizes vitamin D and why you need sunlight to make that happen. But for now, just sort of skim that section about fat-soluble vitamins, look at the structures, get an idea of why they're soluble in organic solvent and in oils and fats, but don't worry about memorizing the structures. There's a second section that is similar about a class of molecules called icosanoids. And I'm not going to talk about these in a lot of detail, but I am going to talk about one specific icosanoid because it's a story that involves research done at BYU. So icosa is the Greek prefix for 20. So icosanoids come from a 20-carbon fatty acid. And that 20-carbon fatty acid is called arachidonic acid. And it has 20 carbons and 4 double bonds, though I'm not going to draw the structure for you. Memorizing it is not important. But this 20-carbon acid is a precursor to lots of different kinds of molecules that are involved in pain and inflammation and thrombosis, which is aggregation of platelets. So the first class of molecules is called the prostaglandins, and these can have different effects. They can lower blood pressure, they can inhibit platelet aggregation, they can control inflammation. If you've ever been in labor, they can stimulate uterine contractions, so not necessarily your friend at the moment. They can cause relaxation of the smooth muscle in the uterus. A second class of molecules is called leukotrienes. These are involved in constricting smooth muscle in the lungs. And so some of the anti-asthma meds target leukotriene function. I'm not sure if they inhibit leukotriene synthesis or inhibit its action, but that's one of the ways that you can help relieve asthma. Another class of molecules is called thromboxanes. Do you need to memorize these functions? No, I haven't. That's why I'm copying them down from my notes. These can trigger platelet aggregation. Platelet aggregation is called thrombosis, and so it makes sense that the thromboxanes would promote platelet aggregation. It can have effects on blood pressure as well. And then there's a last class called prostacyclins, and these dilate blood vessels and also inhibit platelet aggregation. Why this dual role in this class of molecules, both promoting and inhibiting platelet aggregation? Well, it's a delicate balance. You want to prevent blood clotting for the most part, except for when you don't, right? If blood is leaking out of you, you want some blood clotting. But in general, if it's not, you don't want any blood clotting. And so this is the delicate balance there is maintained by the prostaglandins. So I'm going to draw, or by these icosanoids. I'm going to draw you one prostaglandin, and we're going to talk about the story from BYU. This is a 20-carbon molecule, but it looks quite different from this linear 20-carbon fatty acid that's the precursor. You've got a 5-membered ring with sort of 2 tails coming off of it, and frankly, I think you'd be forgiven if you sort of looked at that and thought, hey, that kind of looks like a lipid. I don't know how soluble this actually is in organic solvent. It sort of looks like a phospholipid. I don't actually know if it can. You would expect it to cross the membrane relatively easily. This is prostaglandin F. Oops, that's a PGF2 alpha. What the heck does that mean? I don't know. That's just the name they have given. And if you don't like acronyms for biological names, get ready for biochem, because that's what they do. Anyway, these prostaglandins are, if you think about what has to change with a linear sort of a linear fatty acid that has 4 double bonds, to suddenly have a lot of OH groups and a 5-membered ring, you've got to do some oxidation and some cyclization. And so the enzyme, there's an enzyme that catalyzes this process. It's called cyclooxygenase 1. And because it's involved in oxidation and making the ring. And cyclooxygenase 1 is inhibited by the non-steroidal anti-inflammatory drugs, particularly aspirin and ibuprofen. You can go on the protein data bank, and if you Google, or you don't Google it there, you search for cyclooxygenase 1 in complex with aspirin, and you can see what aspirin does. It actually gets in and blocks a tunnel that the fatty acid needs to get through to get to the active site. At least that's what I think I remember learning. So where does BYU come into all of this? Well, in the late 80s, early 90s, Professor Dan Simmons in our department, he was in the biochemistry area, his lab discovered a second variant of COX-1, which they called COX-2. And this is another cyclooxygenase. And they discovered the gene for it and discovered its function. And then biotech companies used that identified new protein, and they found molecules that could inhibit it. And they generated ultimately molecules that were good at managing pain associated with arthritis and some sort of chronic inflammation. Merck's drug was called Vioxx. Pfizer's drug was called Celebrex. And they found that if you inhibited COX-2's action of generating prostaglandins, that you could address some pain in things like arthritis that were difficult to manage with non-steroidal anti-inflammatory drugs. So this was really billion-dollar big business for Merck and Pfizer until I think the early 2000s, when enough data came in that there were some heart-based, complications to taking these drugs, sort of. And you can read the news for that because I don't remember anything other than that. So some people who were taking the drug developed heart problems, and so they were pulled from the market. So there's an interesting story in intellectual property law, if you want to get into that, with Professor Simmons and the COX-2 enzyme. At the time it was discovered, it wasn't really traditional to patent a gene. And the Human Genome Project wasn't yet done. So BYU didn't patent the gene, though it worked with a small biotech company to try to develop new inhibitors of the enzyme. That company was later bought by Pfizer, but Pfizer didn't believe there was any formal agreement between BYU and that smaller company, and so they didn't pay any royalties or licensing at all to the drugs that had been, based on the drugs that had been discovered by identifying this gene. And so as the proceeds of Celebrex began to skyrocket, BYU filed a lawsuit against Pfizer contesting violation of intellectual property law. The lawsuit dragged on for years and years, and poor Professor Simmons spent a lot of the last few years he was in the department reviewing legal documents, which I'm sure wasn't any fun. But BYU eventually settled with Pfizer. The terms of the settlement were not disclosed, but you can get an idea of what it was by looking at Pfizer's balance sheet during the year that the settlement occurred. They took a substantial write-down in their profits, and you can get an idea of sort of how much came back to BYU as a result. A lot of that money has gone to fund the Simmons Center for Cancer Research. The center was renamed after this settlement. There's an endowed chair on campus with Professor Simmons' name on it. And if you happen to do research in this department, or anywhere really in the College of Physical and Mathematical Sciences, you have the opportunity to have your undergraduate research funded and paid for, so it's like working a job for 10 to 20 hours a week during the semester, and sometimes 40 hours a week over the summer. Quite a bit of money is devoted to that, and that comes from some of the proceeds of the settlement with Pfizer. So it's continuing to enable. Yeah, it keeps the lights on, I guess, and it continues to enable research in our department. So interesting story, but we're really, really grateful for what Professor Simmons did. He taught a biochemistry class when I was here, and I went to him for advice. He's a really, really great person. So anyway, that's the story there. Be prepared to regurgitate that verbatim on an exam. No, please don't. Please don't. But it would be a problem if you had gone through a BYU chemistry experience and not heard about that discovery. Professor Simmons' name is not mentioned in your text, but it should be. Okay, so now we're going to come to the last class of lipid-type molecules that we'll talk about, and this is the part where there is going to be some mechanisms, are going to be some skills that you're going to need to master. Class of molecules called terpenes, and these are involved in steroid and cholesterol synthesis in the body. They also play a role in many, many, many essential oils that can be isolated from plants. As to whether those essential oils have the claimed medicinal effects and properties, the standard is double-blind, randomized, controlled clinical trial, and if they don't have that, they can say whatever they want, but it's not real. Okay? I want you all to remember that. You can, and then a friend once told me that, when it comes to this kind of thing, the placebo effect is real. So if you want to take a sugar pill and feel better about life, you can. Just don't pay a lot for it, okay? So make sure if you're going to rely on the placebo effect to feel better, don't spend a lot of money for it. For example, instead of rubbing some expensive oil between your toes to relieve whatever symptoms, start with vegetable oil that you already have in your house and see what happens, okay? I'm sorry if that offends anybody here. If you are offended by that and would like to come talk to me, I am happy to talk to you about it. I'm sorry to let my own personal opinions color some of what I said. So my apologies. Okay, I'm going to show you some examples of terpenes, and then we're going to talk about their structure and where they come from. So this one, there's a 6-membered ring, and this one is called limonene. And you can tell by looking at the name, this is an oil isolated from citrus peels. And it has, one of the enantiomers has a nice sort of citrus scent, and it's used in cleaning products and flavoring. There are some others that look a lot like it, but are nevertheless quite different. So if you make this structure instead, and I'm not showing you stereochemistry, it is known, but it's not important for our discussion today. This is menthol, which you can isolate from peppermint oil. There's another. It has an aldehyde here and double bonds there, and this is called citral, and it can be isolated from lemongrass, and it seems like it probably has a citrusy kind of scent. Then if you take the alcohol version of citral, you have geraniol, which you can isolate from roses. Here's an even funner one. Are you ready? 6-membered ring. Breathe in and breathe out. Inhale and exhale deeply. And then there's a 4-membered ring fused to it, which looks like that. What the heck? That's the way we draw it looking down from above, though a better drawing of it might be in chair form. These two down carbons would be linked together via a single carbon, and there are 2 methyl groups attached there. Let's see, and then there's a carbon here and a double bond there. So here is the 4-membered ring. And then here is the 6-membered ring. And the 6-membered ring and the 4-membered ring share 2 sides. That's called a fused ring system, a bicyclic ring system. This is called alpha pinene, and it comes from pine trees. If you've smelled the fresh smell of pine wood or pine trees, alpha pinene is a big part of that. It's a component of turpentine. So now, those compounds are all related to each other. And it doesn't look like it, but we're going to talk about how. They have a common precursor, and that common precursor is the following 10-carbon molecule. Oops, sorry. Come on, double bond, double bond. And then you've got what's called a pyrophosphate group on the end. This is called geranyl pyrophosphate, where the pyrophosphate is a really good leaving group. Preview, foreshadowing, it's not foreshadowing if you announce it, I guess. So deliberate preview is this leaving group is at an allyl carbon adjacent to a double bond. So we're going to see SN1 type chemistry here, because if this leaving group leaves, you've got a resonance stabilized carbocation. What's the relationship between these molecules and this one? Well, if you count the number of carbons that are in geranyl phosphate, it's 10. And if you count the number of carbons in all of these molecules, it's 10. We'll do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All of them have 10 carbons. And all of these have a basic subunit that is very useful to recognize, because it will help you generate hypotheses for how these things are synthesized. So the basic subunit is called an isoprene unit. What is isoprene? Well, isoprene itself is this 5-carbon diene with a branching methyl group. If you've been to the Great Smoky Mountains and seen sort of the blue haze, it's composed to a large measure of isoprene, which is coming off the trees that are there, because trees make these terpene molecules, and isoprene is a byproduct of that, or can be made from the similar precursors. We're going to talk about where geranyl pyrophosphate comes from, but right now we need to work on identifying isoprene units. So these isoprene units are the basic sigma framework of all terpenes. And there are going to be other bonds present, but the basic framework is this: 5 carbons with a branching methyl group. And I'm going to choose to number them this way. Carbon 1, 2, and then 3 is the branching methyl group, 4 and 5. Now hold on, because that doesn't follow IUPAC rules, and I don't care. Really, really don't care. If you would prefer to name it a different way, you were welcome to do so. 1, 2, 3 branching methyl group, 4, and 5. So one thing to pay attention, it's useful, as I said, to identify these subunits so you can tell which bonds might have been in the linear precursor and which ones you had to make to go from this linear precursor to these products. So let's try to identify isoprene units in the linear precursor, this geranyl pyrophosphate. And the first thing we're going to do is look for the branching methyl groups. That is a methyl group coming off of a tertiary carbon. So I see one here, and I see another there. The carbon to which the methyl group is attached is carbon 2. And then at this point, when identifying isoprene units, we're going to ignore the double bonds and ignore any oxygens and focus exclusively on the carbons. The second thing we're going to do after identifying all the branching methyl groups is we're going to look for the dimethyl tail of this molecule, because we have at the end a carbon that has 2 methyl groups on it, and we'll call the other methyl group 1. Okay? And then we'll continue numbering: 1, 2, 3, 4, 5, and then carbon 5 of one isoprene unit, and that's our first one, is going to be bonded to carbon 1 of the next isoprene unit in this linear precursor. So now we have 1, 2, 3, 4, and 5, and carbon 5 is attached to the leaving group here. So in pink and orange, respectively, I've identified the 2 isoprene units here. Yeah?
With isoprenes, will they always be connected right together, or can there be like an oxygen or something in between?
Generally, great question. In isoprenes, can an oxygen intervene in this linear skeleton? Usually not. Yeah, I can't think of any examples where that's the case. Now, remember I told you that we're going to do substitution chemistry. We're going to have carbocations. We can add oxygen simply by SN1, right? Water attacks the positive charge. We can move double bonds around by elimination. We remove a proton that's beta to the positive charge, or whatever we want to call it. The sigma framework there may be additional sigma bonds, but we're going to have always a bond between 5 of one unit and 1 of another. All right? So it should be pretty straightforward for you to see where the isoprene units are in geraniol and citral. Let's do limonene, because that's pretty hard. So I will say here that often when finding isoprene units, you will find more than one reasonable way to identify them. As long as your way is reasonable, it's fine. I realize you are uncomfortable with ambiguity, but it's as long as your answer is reasonable, it's good. So let me try to illustrate that. Let's first identify the branching methyl groups. You have one here, we'll call that a carbon 3. And then you have another here, we'll call that a carbon 3. Now I need to look for my dimethyl tail. Where would you say that is on my limonene molecule? And again, ignore the double bonds. Go ahead. Yeah, ignore the double bonds. Focus exclusively on the carbons. Is there a carbon there that's attached to what could have used to be two methyl groups? Okay, some of you are thinking no because you see this double bond there. I tell you, ignore the double bonds to identify the dimethyl tail. Does that make sense? So we're ignoring the fact that there's a double bond here and saying probably this carbon here, these three carbons here, came from this portion of our linear precursor. Yes?
Is that because when the ring comes and attacks at that double bond moves it up?
I will show you the mechanism a little bit later, but in the end what happens is we end up with a carbocation on this carbon here, and we do an E1 style elimination of a proton here to put a double bond there. Yeah. We'll deal with mechanism, probably not today, but next time. So yeah, here is probably my dimethyl tail. I've got branching methyl groups, carbon 2, and I've identified a carbon 1. Anybody want to ask questions about that? Some of you didn't recognize that dimethyl tail. You need to be able to do that. And so if you're struggling to see it, let's figure that out. Okay, so we'll call this carbon 4. Now, there's a couple of ways I could go. I could go this direction and call this carbon 5 and then this carbon 1. And that would work, and then I would say this is the bond I formed. Or, alternatively, and this is the part I think you will hate, I could have gone this direction and called this carbon 5 and this carbon 1. Some of you are saying, buddy, it ain't just this part. So it turns out that either way, you have a carbon 5 bonded to a carbon 1, and 1 is adjacent to 2, which is bonded to 3. So either of those identifying isoprene units is fine. The one that makes the most sense in terms of what the accepted synthetic route is for us to call this carbon 5 and this carbon 1. And usually it doesn't matter if you do it the other way. Usually doing it the other way, you can draw a reasonable mechanism there. So the point is to be able to identify a reasonable connection of carbons in these isoprene units. So once I say this is 5, then this has to be 1, 2, 3, 4, and then 5. So the bond between 1 and 5, this bond in red was in the linear precursor, but there's a new bond that wasn't, and that is one that I have to have made during the process. Okay? So you remember in mechanism problems, it's useful to know what you got to do, what bonds you made versus bonds that were there and didn't change. And this is how you do that. Here are the isoprene units in limonene. Okay. Let's do yes.
Clarification on the dimethyl tail, that comes from the very end of the granule, or from at 1, 2, and 3. Right?
Yep. Yep. Now, I want you, I gave your classmates in the first hour a complicated molecule which we didn't quite get to, but here's the setup. I want you to identify isoprene units in alpha pinene. Okay? You better get good at it because next time you're going to get an even harder one. All right, see you next time.